Hello and a very warm welcome to everyone joining us for today's Select Science webinar titled Elucidating Cost-Effective Miniaturized mRNA-seq Library Construction and Implications for the Study of Circadian Rhythms. My name is Georgina Hughes and I'll be moderating today's presentation. I'm delighted to be joined by our guest speakers, Ryan Martin and Yangping Zhang from the University of Florida, and Dr. Laura Henry from SPT LabTech. In this webinar, we will learn about the metabolic influence of skeletal muscle circadian rhythm on different organismal and disease states, including neurological and metabolic diseases. We will also learn more about automation-enabled RNA-seq assay miniaturization for operational efficiency and cost saving. Following the presentation, we will have time for a question and answer session. If you do have any questions throughout the presentation, please feel free to submit these by typing into the questions box to the right of your screen. Without further delay, I would like to hand over to our speakers, and I would like to thank them for presenting for us today.
All right, so thank you very much for the introduction. I'm happy to be here and eager to talk about some of the RNA sequence strategies that we've utilized, and particularly some of the implications for the use of the study of circadian rhythms, and in our lab, a special focus on the skeletal muscle clock. So today, what I'm going to hopefully talk to you about is to first describe what circadian rhythms are and the underlying molecular clock that drives those rhythms, in addition with a special focus of the skeletal muscle clock. Next, I'll then demonstrate how the skeletal muscle clock impacts our physiology, and then using some of that on how we're then asking some of the implications of circadian rhythms within aging, to which we use a lot of the RNA sequencing tools that you'll hear about later in today's talk. So firstly, circadian rhythms are simply put the physical, mental, and behavioral changes that occur over a 24-hour cycle. And so we see this within ourselves when we are active more during the day and we sleep more at night. And so we commonly use mouse models for this, and we have ways and methods to then monitor their behaviors. So shown here on the right, any black tick you might see is actually the activity of a mouse within a cage. And since mice are mostly nocturnal, we can see most of those activity markers occurring within the night and not so much during the day. Coincidentally, alongside activity, we can also monitor feeding patterns, which is also a circadian behavior, to which most of that occurs during the times that you are awake. And so we see much of the food that a mouse might eat, coincidentally, is at the same time in which they're active. So this simply demonstrates how we might be able to monitor and detect circadian rhythms that then drives our behaviors. Now, simply put, circadian rhythms are then broken into sort of this dichotomy between a central clock and our peripheral clocks that drives these behaviors. Now, the central clock, which resides in the hypothalamus with the suprachiasmatic nucleus, primarily responds to the light and dark cycle. However, all of the other tissues that we have, this stems from the liver, skeletal muscle to lung, have clocks in and of themselves, which we deem those as our peripheral clocks. And so there's this intricate network between all of our tissues and all of our clocks that work synergistically to drive a lot of our behavior across 24 hours. Now, each of these clocks, which are in virtually every single cell that we have, is driven by, on the right here, this transcriptional translational feedback loop, which is primarily characterized by BMAL1 and clock transcription factors that bind to DNA and then transcribe their repressive genes, period, and cryptochrome. Those genes, and finally proteins, translate back into the nucleus and inhibit the transcriptional activity of BMAL1 and clock. Thus, this entire feedback loop operates within 24 hours and is the fundamental reason on why our behaviors change throughout our 24 hours. Now, turning the lens to the skeletal muscle clock, again, at the top here, we see BMAL1 and clock is essentially the fundamental molecular clock that sets the timing of 24 hours. Now, because this is a focus pertaining to a specific tissue, this heterodimer between BMAL1 and clock can interact with tissue-specific transcription factors identified here as MyoD1. And so we have this timing mechanism And shown below, we have other intricate networks within the molecular clock, but fundamentally, these all converge on different points in which this clock mechanism can bind across all different points of our genome. And that then induces the transcription of what we deem as clock output genes. So these are genes that are rhythmically expressed across 24 hours and primarily influence our physiology and homeostasis across that 24-hour period, such as this in skeletal muscle, which influences skeletal muscle's metabolic flexibility, protein turnover, and a lot of the contractile elements necessary for skeletal muscle function. And so not unique to just skeletal muscle, but as you can see on the left here, those number of clock output genes are in all variety of different tissues because each tissue has their own molecular clock. And so in every tissue, you'll be able to detect these rhythmically expressed genes across the day. Now, furthermore, a lot of this work is going to be in mouse models. And so this mechanism is conserved across all walks of life, and particularly human and mouse. And so because mice are nocturnal, if we simply look at the periods in which either humans and mice are active into their rest periods, you can see here by demonstration of the key clock activator of BMAL1 and the repressor period 2 have similar profiles going from the active and rest phase in human and mice. So demonstrating this conservation of this key mechanism that's fundamental to our biology. And so focusing on the skeletal muscle clock, our lab uses this fundamental knockout model of BMAL1 specifically in skeletal muscle. And so this is a tamoxifen inducible under Cre recombinase. And so every skeletal muscle in the mouse lacks BMAL1 and thus does not have a functional skeletal muscle clock. And as you can see, some of the parameters of this induces some metabolic changes to which blood glucose overall seems unchanged with control in the black bar and our knockout in the white. However, when we look at blood insulin, these mice are hyperinsulinemic, and so they have a tremendous increase in blood insulin. And followed by blood glucose tolerance tests, we see that these animals are simply incapable of the uptake and utilization of glucose. And so these So following that, some of the key components associated with it is looking at GLUT4, which is the specific skeletal muscle uptake of glucose. So this key transporter. So on the left, we see that both the mRNA as well as on the right, protein levels of this key uptake protein is substantially reduced after the removal of the skeletal muscle clock. And in addition, we also look at some of the key enzymes that are within the glycolytic pathway, such as hexokinase 2 and phosphofructokinase. And so on the left, we see that the transcriptional expression is reduced, as well as the enzymatic activity on the right, indicating that these animals still cannot uptake glucose and they cannot utilize that carbohydrate downstream. And so this then probed, well, how are they getting this energy from? And so interestingly, what happens is if we look at some of select genes related to the carbohydrate process, many of those genes are downregulated when you no longer have a skeletal muscle clock, indicated in the top bar graph with many of those carbohydrate associated genes. However, below that, in the lipid process, many of those lipid processing genes are upregulated. So now, with the loss of a functional skeletal muscle clock, we have this reduction in carbohydrate uptake and utilization, and more of a compensatory increase in the upregulation of the utilization of fats. And so we really have this disparity of metabolic phenotype when we lose the skeletal muscle clock. And so, as I mentioned before, not only does the molecular clock drive sort of the timing across a 24-hour period, but I to remind you that the clock also binds to a whole host of genes throughout the genome. And so, looking from a circadian temporal analysis with gene expression collected from skeletal muscle every 4 hours, you can see on the left here is a heat map with time on the x-axis and a single gene on the y-axis. And so the yellow representing the peak expression of a single gene, and blue then having the lowest expression. And then you can see this lovely pattern in which some genes are peaking later on, some genes are peaking rather early, but you just see this distinct pattern. And this is a key component of identifying clock output genes. However, on the right heat map, we then have our skeletal muscle knockout mice to which that pattern is completely um, absent. So now we no longer have a skeletal muscle clock, and thus it disrupts this fundamental pattern of clock output genes that then relates to the underlying metabolic physiology that we've seen. And so uniquely here at the University of Florida, we're rather curious about how does the molecular clock is impacting the process of aging, or how might the clock change with aging, which leads me into the focus of today with some of the RNA sequencing techniques that will be touched on, is our recent paper of looking into sort of this aging transcriptome across the circadian time course. So how does the skeletal muscle clock change with age? And to do that, we essentially used young mice of 6 months of age, aged mice of 18 months, and old 27-month-old mice. We then collected tissues of a variety, looked at what are some of the clock output genes in many of these different tissues. And so for today's talk, I'm going to focus solely on the skeletal muscle as an example. And so something that you might get is this, as I've shown before, a very similar heat map of clock output genes. And thus, on the x-axis, we see the every 4 hours of circadian time. And then the y-axis is a single expressed gene. And then we can see this very similar pattern of peak time of expression and then a really reduced time of expression to where it's lowly expressed, demonstrating this 24-hour rhythmic expression profile for a single gene. So interestingly, if you look at each of these three ages, there's 941 genes in the young that reduces down to 699 to 474 in the old. So what does this tell us? It tells us one is that with age, at least in a mouse model, we see that the skeletal muscle clock is still functional. However, the number of clock output genes that occur with age does diminish as we age. And so, as you can further tell from the Venn diagram on the right, many of these clock output and rhythmic genes that we see are rather unique to the age. So they're not variously shared amongst the ages, which was quite interesting. So further pathway analysis, here on the left, we see, well, of all the rhythmically expressed genes, what do they enrich for? What are they doing? And looking at how are they within at least two of the ages. So at the very top, we see insulin receptor signaling. As I've demonstrated, the skeletal muscle clock is rather important for metabolism, as well as circadian rhythm signaling. And so while they're still rhythmic, they are decreasing with age. So the enrichment is not as strong with age. However, lower down, as you can see with AMPK signaling and PI3K, the rhythmicity in those key metabolic signaling pathways is lost. So they're no longer rhythmic in the old. And so while we're still having some clock output genes that might still be there with age, a lot of things are lost or even gained as we age. So shown further on the right, we see that the classifications of all of these rhythmically or clock output genes are primarily in different classes with each age. So for example, in the young, we see more genes that are associated with autophagy and transcription. Compared to the age, we see more proteostasis, some insulin signaling, whereas in the old, we see a large degree of genes associated with muscle structure and some more immune responses as well. So this really demonstrates that, again, there is a functional skeletal muscle clock as we age. The number of those clock output genes does reduce with age, but what those clock output genes is also changing, which is quite remarkable. And so this has resulted in a recent published paper. This was led by Dr. Chris Wolf and Dr. Gutierrez Monreal. And so I encourage you to go out and read that paper, which has some great stuff about different tissues as well, more so than just skeletal muscle. And something interesting that I'd like to highlight as well is that this also led to the formation of a publicly available database known as CircAge, to which all of this data has then been used and deposited here so that you can go in and select which tissues you would like to, and see whether or not it's rhythmic, one at the age and in the tissue to which you're interested in. So for an example, this is a similar output that you might get. And these are some of the prior molecular clock genes that I touched on before. And so you see with 3 different ages, you'll see this rhythmic profile in the tissue, followed by the time on the x-axis and the log2 counts per million on the y. And so with that, just like to highlight a lot of great people in our lab that contributed to this work and a lot of our funding bodies that really helps us dive into some of this. And so with that, I will hand it off.
Ryan just talked about RNA-seq review the relationship between circadian rhythm and aging. Now I'm going to present a cost-effective way for mRNA-seq library construction by use SPT, LabTech, Mosquito, HV Genomics Liquid Handler, and the Dragonfly Discover Reagent Dispensing System. This is outline of my talk today. First, I will briefly show you the workflow of NEB Ultra Directional RNA-seq library preparation, followed by the test of mosquito miniaturization for this application. Next, the experiment design and the result of RNA-seq project by using combined mosquito and the dragonfly library construction method. Then I will show you the individual library trees of one of the Dr. Esser at Dr. Ren's project. After that, the summary and the conclusion. Finally, acknowledgement. So this is a workflow of NEB Ultra 2 RNA-seq library preparation workflow. The first step is to remove ribosome RNA by either polymer enrichment or ribosome depletion. After removal of ribosome RNA, the RNA is fragmented by incubate at a high temperature and followed by first strand cDNA synthesis. Next, the second strand cDNA synthesis occurs, followed by a cleanup. Normally, this is end of day 1 process. At the beginning of day 2, the double-strand DNA molecular are end repaired to create blind phosphorylated ends, and a single adenine overhang is added to 3' end. This enables efficient ligation to adapter with thymine overhangs. The NEB Next adapters are helping loop-shaped adapters. Therefore, we need to use user enzyme to open the helping loop. Next, either a cleanup for small fragment libraries or site selection for large fragment libraries is performed. The final step is PCR amplification of libraries. So at this step, incorporates barcodes to enable multiplex. It also incorporates P5 and P7 sequences required at downstream in sequence process. After cleanup, the library is QC'd and ready to move cluster and generate and sequencing. Before we test the mRNA-seq library miniaturization by SPT LabTech Mosquito HV Genomics Liquid Handler, the question we asked was, can we successfully make libraries with reduced volume reaction use NEB library property kit? Therefore, we manually prepared libraries with reduced volume reactions at scale 1/2, 1/5, 1/10. So the result shows here and in this picture saying that the text input we used is 0, 1, 5, 10, 20, 40, and 100 nanograms. We see at 0 nanogram basically is water control. We don't have any libraries. We have libraries in other input, but so we also don't have libraries for all the test samples. So therefore, and then we did some troubleshooting. We realized that during the beads cleanup, and then the tips touch the beads, and that during the acido wash step, and we basically adding to the first column and take out the acido, and this will making that first column dry if we have a lot of samples. Therefore, we change the protocol by adding acido to all the columns and then remove later. So this will increase usage of tips, however, will increase the efficiency of library construction. We also modified the plate setting and did another calibration of the instrument. After those change, and we did another experiment, and with 10 nanogram, 20 nanogram, 40 nanogram, and 100 nanogram input. And the result shows here is that we are able to get higher library yield and compared to test 1, and the result is more consistent, and but we still have outliers. For optimize this result, we want to have a higher library yield. Therefore, we are thinking logically that this can be done by increase the total RNA input, and it also can be done by increase the PCR amplification cycle numbers. However, the increase the PCR amplification cycle numbers will cause PCR duplicates. Therefore, we decide to using 200 nanograms and at 1/5 scale move on forward. The reason is that logically speaking, because the maximum input of this kit is 1000 nanogram. So therefore, logically speaking, the maximum input for 1/10 reaction should be 100 nanograms. That's why we choose 1/5 reduce reactions, move on forward. So a few months later, we also request the SPT LabTech Dragonfly instrument. So with this instrument, we want to incorporate the Dragonfly into this protocol. Therefore, we conduct another test run. This time, we use the customer sample. The expert design is like this. So we use the same 4 samples, and we use the same input, and we process manually at full scale, and with combined mosquito dragonfly at 1/5 reduce the volume reaction. We also process for sequencing and the data analyze. This is the final individual library trace on tape station. The sample ID 124 is mRNA library prep by mosquito and the dragonfly, and 1/5 reduce the volume reaction. 5 to 8 are total RNA-seq library preparation by mosquito and the dragonfly at 1/5 reduced reaction. 9 to 12 are mRNA library. 13 to 16 are total RNA libraries prepared manually at full scale. As showing here, and the result indicating combined with mosquito and the dragonfly, RNA-seq library construction are pretty consistent compared to manual preparation at full scale. So this is the data analyze, and as the sample ID shows here, and then 1, 2 are same cell line, biological replicates. 3 and 4 are same cell line, and biological replicates. From this figure, it shows in both cell lines the data between manual preparation at full scale and the 1/5 reduced volume reaction with mosquito and dragonfly are consistent with R squared about 0.95. This is the correlation of total RNA-seq data between manual preparation at full scale and 1/5 reduced volume reaction with combined of mosquito and dragonfly preparation. Same as mRNA-seq data, the correlation between manual at full scale versus 1/5 reduced volume reaction with combined of mosquito and dragonfly are similar. The R squared is about 0.85. The feedback from our customer is that the gene count of mRNA and the total RNA are similar as well, with R squared of 0.83. The data is not showing here. This is the example of individual library trees for Dr. Esser and Martin's project. For this project, there are a total of 33 samples. This shows the library preparation is pretty consistent. So this is the sequencing data and the raw sequence number. And basically, for this project, we get average of 57 million reads per sample, and the lowest number is 15 million reads, and the highest number is 68 million reads. So with this, the take-home message are, library construction is more consistent, use combined of SPT LabTech mosquito HV genome liquid handler and the dragonfly discover preparation. As the data showed, the technical replicates for each cell lines are quite similar, with R squared higher than 0.95, and the mosquito dragonfly versus manual are similar, with R squared is about 0.85. The poly A and the ribo gene counts are similar. The R squared is 0.83. SPT LabTech mosquito HV genomics liquid handler can reduce the reagent cost. It can process 384 samples simultaneously, thus increase the efficiency. There are faster turnaround time because 384 samples processed simultaneously. This can help customer for their time-constrained projects. Finally, I would like to give thanks to Brendan, Core Facility Specialist 2 at the ICBR Gene Expression, who did all the hands-on work. ICBR NextGen team processed the sequencing run, and ICBR Associate Director for Science, Dr. Steve Mador, and ICBR Director, Dr. Annalisa Paul, for their support of the pilot study. I also want to give thanks to Dr. Mingyi Xie, Associate Professor at UF College of Medicine, for providing a sample for testing run. Last but not least, Dr. Esser and Dr. Martin for involved us in their project. Thank you for giving me the chance to present this work.
Thank you everyone for that informative presentation. So it's now time for our question and answer session. So if you do have any questions, you can still send those in now. You can submit these by typing into the questions box to the right of your screen. So to kick off our Q&A today, my first question here is, can every kit be used with the SPT LabTech mosquito and dragonfly system?
I go ahead and answer this question. Technically speaking, yes. And first, we need to test in the kit whether they are able to successfully get the libraries and by reduced reaction while you use that kit.
Yeah, and to add on to that, we've looked at over 40 kits for miniaturization and with both the mosquito and the dragonfly. And we are able to reduce or miniaturize these kits typically around 5 to 10x, but there's some that can even be used up to 25x as well.
Great, thank you. So we'll move on to our next question, which is it hard to write the scripts to set up a new protocol?
So I can answer this question as well. So, and from our experience and the mosquito, dragonfly, liquid handler, they're very user-friendly and it's not so hard to write the protocols.
Great, thank you. What other next-gen library construction protocols have you implemented in using the mosquito and dragonfly system?
So far in our core, so we did a COVID-seq, we did a DNA-seq, mRNA-seq, and in 16S, as Laura mentioned that there should be more than 20 kit, and then from their tech support group.
Yeah, so actually on our website, we even have a full list of kits that have been miniaturized either by us or by collaborators. And what's included in on the website is any papers that those have been used in, or any webinars that they've been mentioned in. And then there's also on our newest edition of our software, we actually have wizards for some of the most common protocols. So we have, the NEB Next Ultra 2 protocols for both DNA and RNA. And then we also have some of the DNA quantification protocols as well. And when you have your mosquito and or dragonfly, that's something that we work with customers with our field application scientists to really optimize those for what you guys are working on.
Great, thank you. What behaviors might influence our circadian rhythms, i.e. molecular clocks?
I'll be happy to answer that one. Many different behaviors actually can. Perhaps the most relatable might be if you've ever taken a flight and have landed in a different time zone, you probably don't feel all that great for some time after that. So certainly jet lag can be part of that. So simply put, the light and dark cycle. So if you think about going into a different time zone as well as shift work, so the light and dark cycle is a tremendous cue for our circadian rhythms. And then beyond that, we also have different cues such as feeding. So different windows of consuming food as well as physical activity and exercise, which can more or less have more of an effect on some of our peripheral tissues. And so it's really important to have those in sync most of the time. And so any number of those cues that will essentially manipulate your molecular clock can then manipulate your downstream physiology.
Thank you. What methodological approaches or considerations might be needed to detect oscillating genes?
So some of the biggest considerations would certainly be the temporal aspect. Typically, you want to use about every 4 hours, certainly with a number of samples within each of those time points. So for example, if you only use, say every 6 hours or even at 12 to 12, you're not necessarily going to be capturing any rhythmic expression of those genes. So you need to have it close enough if you're wanting to detect any type of rhythmic pattern for the specific expression of a gene. In addition to that, also being mindful of how you're collecting your samples as well. So sometimes even a small ounce of light that might react to, let's say, an animal, for instance, can really manipulate the clock as much, that is not going to be all that advantageous. Another consideration would be to, such as in the paper that we have published, is the consideration of constant darkness. So a lot of our molecular clocks are self-sustaining, meaning they do not need any type of signal to keep going. And so if you truly want to see the rhythmic genes of a tissue, you're then going to put that animal, for instance, in constant darkness, to which they don't have a light and dark cycle, but yet their molecular clocks are able to function on their own just fine. And so that'll give you a better readout of some of the oscillating genes in each of your tissues.
Thank you, Ryan. And I think we'll move on to our last question for today, which is, what were the RIN values of the samples that you were working with? And what level of miniaturization did you carry out for the poly A selection and rRNA depletion?
So I go ahead and answer that question. So to get a good data, reliable data, we always suggest to start RNA. Basically, is RNA integrated number higher than 7. And what's the second part of the question? I'm sorry.
I'll repeat the second part of the question. What level of miniaturization did you carry out for the poly A selection and rRNA depletion?
We carry out 1 to 5, and basically is depends on the capacity of the kit and the best result. We be able to do with 1/10, but carry on, move forward, and then regularly we do 1/5.
Great, thank you. And I think we'll leave it there for today. So I'd like to thank all of our expert speakers once again for the informative presentation and discussion, and a big thank you to everyone who joined us online. We hope that you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speakers. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.